How are you all? Welcome to our fresh channel Biotech Mirror. COVID-19, pandemic, new normal, testing, etc. You must be hearing this word almost every day. So now what is COVID-19? COVID-19 is a pandemic caused by a virus called SARS-CoV-2. But how it is detected? Here comes the word testing. So let's discuss some facts regarding testing of COVID-19. In India, all testing guidelines are provided by the ICMR, Indian Council of Medical Research. In India, there are total 4 strategies which have been approved by ICMR. These are 1. RT-PCR testing, 2nd. Antigen testing, 3rd. Antibody testing, and 4th is TrueNet, also known as CBNet testing. You must have heard about RT-PCR testing. Now, what is that? And is there any other strategies which have been approved by ICMR? Let's find out. In today's video, we will be talking about all these strategies. RT-PCR testing. The full name is the real-time polymerase chain reaction test. Is most commonly used one for COVID-19. It is primarily based on a PCR. Now, PCR is a process that repeatedly copies and amplifies the specific genetic fragments of the virus, ensuring that there is enough of a sample to conduct the analysis. The RT-PCR test starts with a simple swab taken from inside a person's throat or nose. Coronaviruses have RNA or ribonucleic acid as the genetic material. However, the swabs from the patient yield only a tiny quantity of RNA, which is not adequate for the testing process. To overcome this problem, the RNA as single-stranded molecule is converted into a double-stranded molecule using an enzyme. Now, this process, patient samples, primers, and probes are together left in the PCR machine for the binding process to take place, and a fluorescent signal marks the presence of the virus. Now, the time duration for the testing process is around 4 to 8 hours, but results may take up to a day to arrive because of the time taken to transfer the sample from the sample collection units to the labs. The RT-PCR test is also the most expensive of all those tests used for COVID-19. Initially, the cost of the test was capped somewhere between 4,500 to 5,000 by the ICMR. However, the cap was recently removed and the state government were asked to fix the cost of per test accordingly. Now we come to our second test. It's called antigen testing. A new antigen test that has been approved for COVID-19 diagnosis in India give result in only 30 minutes. Now, if we talk about the tests are designed to detect a specific protein present in the virus, and that may draw out the body's immune response. In this case of COVID-19, it is the spike protein that is present on the surface of the coronavirus that facilitates its entry to human cell. For this test, professionals collect a nasal swab, which is then immersed in a solution that deactivates the virus. Now, a few drops of this solution and then put on a test strip. This has to be done within an hour of the immersion of the swab in the solution. A few drops of this solution are then put on a test strip. This has to be done within an hour of the immersion of the swab in the solution. The test strips contain artificially antibodies designed to bind to specific COVID-19 proteins. If a person is infected with coronavirus, the test line will appear on the paper strip within 15 minutes. Since the antigen test does not involve any amplification process, the swab sample may lack enough antigen material to be detected. This may result in false negative test, and this is the reason if a person tests negative through antigen testing, they still need to get an RT-PCR test done for confirmation. If a person tests positive, however, a confirmation RT-PCR test is not required. The advantage of using this test is that it reduces the burden on relying just on RT-PCR test to identify COVID-19 patients. So, let's talk about the third test in our series, the antibody test. Antibody test, also sometimes known as serological test, detects whether a person has antibodies to the virus. Antibodies are naturally produced by the body's immune system to fight off infections. Antibody test cannot be used as diagnosis for COVID-19, but can reveal whether a person was recently exposed to the virus. Persons with antibodies in their bloodstream are likely to have immunity to the disease, although scientists are still not sure how long COVID-19 antibodies offer protection from the infection. For the antibody test, trained professionals collect a few drops of blood. The sample is placed on a cassette or cartridge that contains the SARS-CoV-2 protein. If the blood sample contains the antibodies, they will immediately bind with the viral protein, showing the positive results, as it has been indicated just like a home pregnancy test kit. Antibody test can be of different types. One of these tests, known as enzyme link immunosorbent assay, also known as ELISA, which is designed specifically for screening large number of specimens at a single time. It is suitable for detecting the spread of a disease in a very large population within a short span of time. Antibody test can be useful to carry out surveys to check whether a population has been exposed to the virus or not, etc. These are currently only being used for research and surveillance purposes. The TrueNet test. On 19th May 2020, the ICMR revised the testing guidelines, and in these guidelines, they allow the use of TrueNet system for screening and confirmation of COVID-19. What happens here is basically it's a chip-based test worked by detecting the SARS-CoV-2 specific E gene, which helps bind and developing the envelope that holds the compounds of coronavirus together, and the gene for the RNA dependent RNA polymerase, then an enzyme that enables the virus to replicate. Unlike the traditional RT-PCR, the sample preparation in TrueNet test is automated, and the results are available within half an hour to 1 hour. The test use generally nose or throat swab samples, which are collected from patient and dipped in a solution that inactivates the virus. A few drops of the solution are then placed on the cartridge. On the inserting the cartridge into a machine, a pre-programmed reaction is initiated, which extracts the nucleic acid or the genetic material from the sample. This has to be followed by an RT-PCR. Why? Because the purification nucleic acid is added to a microtube containing freeze-dried RT-PCR reagent, and the solution is allowed to stand for about a minute. The solution is then applied to a microchip, and the test is inserted into another machine, which then reverses the transcription and PCR takes place. The advantage of this test is that it is very quick and portable. This allows teams to set up mobile testing centers or kiosks in certain containment zones instead of having the samples transported to the labs. TrueNet is an indigenous developed portable version of CBNet or cartridge-based nucleic acid amplification test, also sometimes known as Gene Expert test. Both the tests were initially developed for testing tuberculosis. While both versions are cheaper and quicker than RT-PCR test, TrueNet is battery operated and portable, while CBNet machine needs an uninterrupted power system and air conditioning, and hence cannot be deployed at automated and containment zones. TrueNet testing kits are something priced of around 1,200 rupees per sample. Each kind of test that we discussed today has different process, prices, and also purposes. Antigen test can identify positive cases, while antibody test can screen a wide area. The testing figures that ICMR updates every day include data only from RT-PCR and TrueNet test. Neither antibody test, which are for surveillance purpose only, nor the newly introduced antigen test are included in the ICMR data. So that's all for today, friends. We will keep bringing information for you, so stay connected. Bye.